let's go ahead then to today's material. And okay, again, I've collapsed the first 13 weeks, and I've also put everything back where it was supposed to be. So, we move things into week 13 just before the test. What we're going to start on today is, well, practicum 3 was part of this week. Okay. So now we're going to start. We won't finish. I'm going to take the portion we don't finish here and push it into week 15 and change the dates. It's no problem. We're going to look at organ systems. You've studied organs all along. You have done your reading all along. And much of the information in this is going to be repeat for you. Well, that's good. You got 3 more tests, and you don't want them to get harder. They do tend to get harder, however, if somebody doesn't sort of pay attention to what's going on. Organ systems, and there's a handout I have for you. Organs and systems in the lab. Hmm. And here it is next week, finishing off. Okay. And tissues. Tissues. There's 2 parts to the tissues. So I don't see the second part. It's okay. We'll just do the first part today anyway. Tissues is histology. And while you've been reading, you've seen photomicrographs, you've seen tissue sections, uh, and for the most part, they were to be, most of them were to be skipped. Some of them weren't, but most of them were. And we need to know these tissues and then come back to the organ systems. And the histology portion tends to, or the tissue portion, where I've got the pointer, okay, tends to be one of those topics where if a student catches on to it right away, okay, or goes home and or is home and reads about it, uh, then they usually catch on and then they're going to do fine. We've got a lot of practice material for this for you, so that you'll be able to hopefully do well on the practice. But for some students, when they start histology, they just like throw up their hands and say, I can't get this. I can't get this. You do that and you won't get this. Don't don't give in. Don't give up. So what? If it's a little bit different. You're gonna want to catch on to it pretty soon. Okay, now first the reading assignment is chapter 4. Chapter 4 is tissues. Okay? It's the entire chapter. I'm not going to go through it with you. I don't need to for an entire chapter. Okay? Okay. It says the assignment's due November 23rd at midnight. Frankly, it can be turned in anytime before P4, practicum 4. Um, Again, I don't like to use the word late. Okay, so let's see. I'm starting on today. No, that's not today, is it? I want to get rid of this. Sorry. Okay, 1119 tissues. Great. This is where I want it to be. There are four types of tissues, or four tissue categories. Actually, there's many subcategories. We've talked about these before. So again, if something sounds like it's a repeat, it probably is. Okay, just to be sure that you've got it down. The four basic categories of tissue are right here. Epithelial tissue is tissue that covers or lines all the linings of the blood vessels, all the linings of the GI tract, the outermost layer of the skin. Those are epithelial tissues. Connective tissues interconnect to other tissues, store fat, insulate, or they can cushion. And then there's muscle and nervous tissue. On epithelial tissue, these right now uh, in the laboratory are the hardest for people to comprehend. I I don't see it. Uh, I know it doesn't give everybody a bad time, but if there's any part to pay attention to, it's going to be right now when we're talking about the epithelial tissues. These tissues are categorized by shape and arrangement of their cells, two basic characteristics. The shapes, the basic shapes are cuboid, and you can see this first diagram. These are cubes, okay? They look like a whole bunch of dice stacked next to each other. Okay? So cuboid is one shape. Another shape is columnar, and we'll have to wait till we can get to there. And the third shape is squamous, flat as a pancake. Squamous, we had that term when we talked about the temporal bone. Squamous really doesn't mean flat, but that's the description you would give anyway. Flat. Squamous actually means like the scale of a fish. Well, that's flat. So we have three basic shapes: cuboid, columnar, and squamous. And if they're in a single layer, like this one, just one single layer, sort of like brownies that you have cooked and now cut, and there's a single layer. This is called a simple epithelium. If there are two layers in height, then it's a compound epithelium. And if it's many layers in height, it's a stratified epithelium. Simple, compound, stratified. Well, these guys have to stick to something. They just don't float around in your body. They may be the inside lining of the small intestine, for instance. No, they wouldn't be, because that's not simple cuboidal. They may be the inside lining of the duct of the kidney. A kidney tubule would have this kind of arrangement. And this single layer is attached by a basement membrane. That's a catch-all. The basement membrane is a type of connective tissue that abuts onto an epithelium. How it's composed or what it's composed of, there are so many variations. We're not going into those. Just know that the basement membrane is where this attaches. Sort of like if you imagine a zillion here, false teeth, and here's polydent. And the polydent serves as the basement membrane that they're adhering to. In simple epithelia, like this one, simple cuboidal epithelium, we're naming it based upon how many, its arrangement, one layer, its shape, cuboid, and the fact that it is an epithelium, that it coats something or covers something. I'm just letting this creep in here. This is a kidney tubule, and it's cut transversely coming out towards you. And you're going to see the cells that go around it. And you're going to tell me, wait, those aren't cuboid. Those are more columnar. In this particular picture, which was a poor choice, I think, they do look that way. But we look over here at the diagram, and the diagram shows what really is the case in the surrounding tubules. Here are the epithelial cells covering or lining. These are lining. And they are essentially cuboid with the nucleus almost dead center. How do you get this round shape from this flat shape? You take the two ends here and here and roll them up like you would roll up a piece of crepe paper, not crepe paper, but cardboard or newspaper to make a roll out of it. And that's what's happened here. Where's the basement membrane? They haven't labeled it. Oh, yes, they have. They're calling it basal lamina. The basement membrane is on the outside. Because when you open this back up, if you were to cut it and fold it flat, you'd get this. Conduction, moving something from one spot or location to another, is the main function of simple cuboidal epithelium. Okay. And the best place to see it is in the kidney tubules, as in the loop of Henle that we talked about before. Okay. In the cortex of the kidney, we find areas like this, little piece of one here. These are called glomeruli or glomerulus. These are the kidney tubules. The kidney tubules, magnified. I'm going to come down here. Here are the tubules. Here's the epithelium. It's a single layer. If it looks like two to you, one, two, that's because this one belongs to the tubule over here. They're just touching each other. A single layer of cuboid cells, simple cuboidal epithelium. Whenever you hear that, you want to probably immediately think kidney tubule, kidney tubule. And a little bit later, when you get to the practical exam, there'll be 33 PowerPoints of tissues and organs, and this might be one of them. And you need to recognize it as kidney. Okay, we'll talk about the glomerulus in a moment, but it's kidney. These are kidney tubules. These are simple cuboidal. Reading chapter 4 is going to help you, don't worry. Okay. Cuboidal epithelium. The second type of epithelium, then, let's do simple squamous. Simple meaning one layer. Squamous meaning, well, let's call it flat. Where are we going to find this? We're going to find it along with the glomerulus, outside of the glomerulus in Bowman's capsule. This is a magnified glomerulus. The blood is coming into Bowman's capsule in the cortex of the kidney through the afferent arteriole. Okay, it comes in here. You can see the red blood cells, and it will move throughout the glomerulus itself. Since the blood's under pressure, plasma is being ejected, and that's why there's always a space here, at least under the microscope. There's always what looks like a space. That's the glomerular space. Well, something's got to contain this, and here it is right here. And it is thin, one layer thick. These represent nuclei. Here's a nucleus here. The cell is here. It's one cell thick. Can you imagine if you have high blood pressure, how easy and unfortunate it would be to pop these things? It happens. Okay, Bowman's capsule here, a good example, is simple and squamous. So it's simple squamous epithelium. We do have other examples of simple squamous epithelium, the lungs being one of those. When we get to the lung slide, I'll be sure to point it out to you again. Now, simple columnar. Still working with simple, one layer, but now we've turned to our third shape. We did simple, cuboidal, squamous, and now columnar. Anybody? Oh, you probably read ahead. So I was going to ask anybody know where this picture would have been taken from. It's taken from the small intestine. These are villi. Okay. Just turning the picture upside down like this throws some people. Get over that. Okay. If you ever stand on your head when you were a kid and watch television, you can do it. Okay. This outer layer here. This is simple epithelium because it's only one layer thick. The nuclei tend to be towards the base of the cells. And the shape of the cell, including the nucleus and the cytoplasm, really good over here, is columnar, except for the upper esophagus. Everything else from the lower esophagus onward in the GI tract is lined by simple columnar epithelium. So if I show you large intestine later on, if I show you sigmoid colon later on, small intestine later on, it's all lined by simple columnar epithelium. This is primarily used in absorption. It's absorbing nutrients and some water across itself, and it goes to the center. Now, the center, all we can see here is the lamina propria. Lamina propria. There will be a lacteal, but I don't see one in here. The lamina propria is pretty obvious. Compound cuboidal. I don't cover this. I can mention it. I can tell you where you're going to find it. It's 2 layers of cuboidal cells turned into a roll or a tube, and you will find it in the ducts of the sweat gland. That having been said, if you want to look it up again, you can, or you can ask me about it, but I will not test you on compound cuboidal. Nor will I test you on compound columnar. These are stacks of 2 columnar cells, 2 layers of columnar cells. And these are found in the ducts of the salivary glands. But I'm not going to ask you that. Okay, so stratified squamous. We've already talked about this. You've got it in your notes, okay, and you will be tested on it on lecture exam 3. That's the integumentary system. Okay. If you recall, when we went over it, the integumentary system starts at the bottom here at the stratum basale. The stratum basale is attached to the basement membrane. None of the other layers are. The stratum basale or stratum germinativum produces the next layer, the stratum spinosum. And as more and more cells are produced and pushed forward, they change due to pressure and due to the fact that they're coming to more contact with ultraviolet light. The next layer is the stratum granulosum, stratum lucidum, and the stratum corneum, which contains a lot of keratin, which stops you from drying out. The epithelium, or of the epidermis that we have here, is many layers thick. Many, not just one. That would be simple. Not just 2, that would be compound, but many. So it's stratified. And the final shape of the cells before they're lost is flattened. They didn't start flattened, but they ended up flat. And that's what we look for, the final shape. So this is stratified squamous epithelium. Transitional epithelium. Transitional means that something can change its shape. Okay, it can morph. Transitional epithelium stretches. It can align a tubule, and when something passes through or accumulates, the transitional epithelium can stretch to allow or accommodate the extra material. This is a multi-layered epithelium. So you might want to tend to call it stratified, but we'll see why we don't. Lining the ureter is the most common place to find transitional epithelium. Here's the epithelium. It's folded in. If you can imagine a circle of this epithelium going all the way around and more coming in, and I do have a picture of that, then you see that it is multi-layered. But when something is multi-layered and it's an epithelial tissue, it can only be one of two things. The stratified squamous that makes up the epidermis that we just saw, or it's transitional. How do we know the difference? The stratified squamous has flat cells at the end. This does not. These cells can be any shape. Some will be flat, but this image, everything is sort of rounded and cuboid, but it's multi-layered. So this is transitional epithelium. Here's a long name, pseudostratified ciliated columnar epithelium. Okay, epithelium is going to line something with columnar cells that have hairs or cilia. And then there's this word pseudo-stratified. It's going to look to be stratified, many layers thick, but it is not. It's only one layer thick. This is a single layer of epithelial tissue that gives the appearance of two or more layers under the microscope. Imagine for just a second that you're in, well, I think I was in kindergarten or first grade. They take a school picture. They got all the kids out on the lawn, and they got the photographer there. And everybody lined up, and the photographer immediately said, "No, there's too many of you. It's too wide. Double up." And so we doubled up, but it was still too wide. So we tripled up, but that left kids standing in front of kids. So the first row of kids sat down on the grass. The second row of kids sort of squatted over their shoulder, and the third row of kids remained standing. So you've got everybody's head in the photograph, but it's only one layer of kids because every kid is standing on the grass or sitting on the grass. Here's pseudostratified ciliated columnar epithelium. Well, columnar, I can see a lot of columnar cells right away. Okay. I can see cilia up here. It's a good picture. And pseudostratified means it looks like there's two layers or so. Here's one, this one, maybe three here. But really, they all touch the basement membrane. This one is coming over from the backside of this one. Okay? It's leaning over, leaning in. Here's the kid sitting on the grass. Here's the other kid leaning over his shoulder. Okay? And you could have some kids standing up too. These are not on top of each other. They are pseudo-stratified. They're not stratified at all. We have defined this in the respiratory system. We need these cilia to beat and constantly get mucus out and dust out and any other particles that are not supposed to be in our respiratory system. And that includes not only the lungs and the tubules of the lungs, but the nasal cavity as well. We will find the pseudostratified ciliated columnar epithelium, this picture, also in the larynx and the trachea. Okay. I'm not asking you to turn this into me. I am suggesting strongly that you be able to do it. Okay. Here's the name of the tissue that we had, minus the two that I said I'm not going to bother with. Simple cuboidal. Find it in the text, probably in chapter 4. Find it. Put the text page down for yourself so you can always go back and look at it again. But you're going to need to know approximate function and where you would find it in the body. Simple squamous, well, textbook page, I don't know it either right now. You'll have to look that up. Function contains the glomerular fluid. Location, Bowman's capsule. Simple columnar, find a good text page. Simple columnar, function, absorption. You can add to that of nutrients, location, small intestine, or you can say GI tract, except for upper esophagus. Okay, now, any questions so far?
Nope.
Does it look impossible?
No.
Not if you take it a little at a time and keep up with it each day. I think you'll do quite well. But if you get behind on this stuff's not easy to catch up on. Okay. The uh, well,
Charlie, I have a question.
yeah, go ahead.
You said a discussion will be held in class today, or is there no
We're
discussion at all?
doing it right now.
Okay,
We
cool.
have been doing it. Okay. I don't need anything turned in. Okay. There's going to be one more chapter heading to read, and that's the reproductive system, and that'll be the final chapter that you'll outline. But as far as discussions, uh, uh, I, it, I sort of beat my brains out trying to figure out a discussion topic that I like, and I thought that maybe you would like. If you've got a discussion topic, send it in to me, and I can give you credit for it, or we can go anonymous, and I can throw it out. I do have some things I would like to talk about, but some of them I know are sensitive to people who may have had a death in the family or something like that, uh, and it stops me from going there. But if you would toss them my way, uh, I'd appreciate it. If there's one that you were expecting, I'd appreciate it. And okay, but there's not going to be any more written uh, discussions. Okay, here I'm back at the modules again. Okay, and here's the tissue lecture that we just did right here. That's only one of them. There's more. And then below that, P4 handout for the organ systems that are coming up. You've done all of these organ systems one way or another, either by reading them or we've actually studied them For instance, I'll just mention the kidney. How many times have we done that one? And yet it's going to be subject material for P4. That's the way they put this together. That's fine. That should be easy for you to handle. For the head region, we've covered the parotid gland, Stensen's ducts, and maxillary. We did not do the vestibule yet. Okay, but I will be doing these starting on Tuesday. And is this blue?
Yeah.
Okay, in blue, I added to the parotid gland what type of tissue makes it up. And all glands are made up of glandular epithelium. What could be easier than that? You know, I wish everything in this portion were, but it's not going to be, unfortunately. So when you need to know something, I put it here in blue. The palatine tonsil. Well, it's a type of tissue we haven't gone over yet, reticular tissue. The epiglottis. You know what the epiglottis is, but may not know that it's the elastic cartilage. Anyway, I put it in blue. Here's the respiratory system that we're going to be going over. A lot of the terms, like right here, you've seen them before. That doesn't mean P4 is going to be a total rehash at all. It's a test of its own. Okay. Again, purple and stuff for the abdominal region. We'll go over the liver. The reproductive organs. Almost everything here is glandular epithelium. The integumentary system that we have already gone over. The epidermis is stratified squamous. And the dermis underneath it, we'll describe this on Tuesday, is dense irregular connective tissue. The endocrine system, these are all glands, so they're all composed of glandular epithelium. You'll need to be able to identify these on models. Okay. All of these have models associated with them. Okay, and I have those for you in the material that you'll be given. Now the kidney lecture from way back has all of this stuff. Okay, and it has the different types of tissue types, mostly simple cuboidal and the transitional epithelium in the ureter. Okay, on Tuesday, okay, if you recall, uh, yeah, right now I'm looking at a bunch of black tiles with your names on them. Okay, I'm going to need you to, yeah, I'm going to need you to show yourself, show your face, for most of you. Yeah, if you're very, very shy, I won't insist. You're not getting graded or anything. I just need some help with the guys that's coming in to evaluate me. I have the feeling that there are two people in this world that want to get this evaluation over, me and him. He has to come and sit for 50 minutes with us. That's okay. We can do that. We let him come in at the start. I don't know if I'll introduce him or not. If he comes in with a black tile, I'm going to make him show his face because he's having us do it. And I'd appreciate it if you did great today, but keep it up. I like to have a back and forth between us, answering and asking questions. Uh, I don't know if I can tell a story. uh,
Wait, Charlie, is this next meeting?
yep.
Okay.
Yeah, it's next meeting. It's and then we'll be done with this. I mean, after 45 years, I've done enough of these evaluations. Leave me alone. But it does. It's a little tiny bit nerve wracking to have somebody looking, over your shoulder like that. But I'm not going to care much. But I would appreciate the help of a couple of questions from the reading this weekend, or anything at all. Questions about the practicum, uh, questions about the lecture test coming up, question about how are we going to do all three tests in the short time remaining. Generally, if I had a class, they would be asking me, what are we doing after the class is over? Where are we going? How are we going to celebrate? Huh. I don't think, unfortunately, this is the time for that, but it used to be. I was not always, in case you think I was not always a stick in the mud. I used to have lots of parties. I used to live out in the canyon. I'd invite all of my classes and all of the instructors. I didn't care. Everybody could come on out there. It was not on school time. And usually my parties lasted 2 days. Uh, we would, they could bring sleeping bags. They could bring sleeping bags if they wanted to. They could use the house if they wanted to. They could go down in the root cellar and sleep if they wanted to. Stream in the backyard. They could pitch a tent if they wanted to, so that they could manage to stay the 2 days. Some of them had to stay because they were in no condition to leave. And I never had to leave because it was my place usually. Once we had about 60 people there, I was in the backyard along a retaining wall. It's a short wall to hold the soil up, keep it out of the stream. There were about 10 people on that retaining wall with me. And we had a bonfire going. Yes, I like bonfires. And I was playing a guitar. Didn't take up the banjo till later. I was playing the guitar and feeling no pain. And in the darkness, I leaned over backwards from the retaining wall and fell backwards the 8 feet down to the rocks in the stream below and knocked myself out. Yeah, knocked myself out pretty good. And when I came to, I could see the guitar away from me. I could hear water running through my ears because that's how deep the stream was at that time, just very, very shallow. And I'm lying down looking at the stars and sycamore trees. And people are running around. Where's Charlie? Where's Charlie? They didn't even see me fall off. It's like you have a row of teeth and one of them suddenly is missing and you don't know it. Well,
That's
they
awful.
some girl in the class up on the ridge looked down and saw me finally. I couldn't move. My shoulder was not busted, but it could have been. My head was really messed up, and my arm, left arm. Well, anyway, they called the fire department. The fire department is volunteers, and that's my neighbors, and they raced right by my house. Then they found out where it was, and they came back, and they pulled me out of there on a stretcher. Uh, I'm feeling a little bit weird. People are saying, "Is it okay if we could go ahead and party?" And I said, "Yes." And two ambulances had arrived. One came from Orange. One came from Orange. One came from El Toro. And they were arguing about who got to take me because they came all that way out there. And I said, well, most of my friends that I have, the people I know, are in the El Toro area, not out in Orange. So I guess that's where we'll go. We'll have to go to the death hospital. The death hospital is Saddleback Memorial. Uh, uh, Not my favorite place to go. Not my favorite place to go at all. Anyway, two guys followed. They put me in the ambulance and took me into El Toro. Two guys in the class followed. They were in a small Volkswagen Bug. And I don't know why they were coming after me. I guess they were going to see what's going on. And I'm inside of the treatment room, and they're stitching up my head. I'm on my stomach, and they're stitching up my head, and it's like shoelace. And every time they pull, my head would come up, and there's a window in front, an observation window. And here's the two guys, two students, and they're going, "Yeah," and the head goes up, "Yeah." They kept it up on me all the time, thumbs up, and uh, I got a lecture from the doctor about my morals. My morals are fine, you know. I just did something dumb, and those two guys took me and helped me. I still had anesthetic on me, shot into me for a little while, not long. They put me in the car. They put me in their Volkswagen. They pushed me in the back seat. Did you ever see Weekend at Bernie's? Okay, where they drive around with the dead guy in the Volkswagen? That's what it felt like. I was really pressed in there, and now I'm getting pain. And they say, "Where do you want to go?" I said, "Well, I don't want to go back to the party. There's no way. They can leave when they want." I said, "Drive me into San Juan to my girlfriend's house." She has her own house. She'd been at the party earlier, but she was smart enough to leave before it got typically the way it gets. And so they drove me into San Juan Capistrano and stopped about 4 in the morning now. And they pulled me out of the car. I mean, the pain is terrible now. And I'm walking up to the door, and the light goes on, and the door flings open, and I don't know what's going on, but all of a sudden, smack, somebody hit me in the face. Somebody slugged me in the face. Then somebody slugged me in the chest. And then somebody kicked me. I was getting beat up, and there's nothing I could do about it. I could hardly move. The two guys pulled me away, fortunately. I got back in the car and I looked, what the hell happened here? And I said, "Oh no, that's not my girlfriend's house. That's my ex-girlfriend's house." And I'd taken them to the wrong one. Oh God. So that was the last one of those parties I've had. Okay. Now, uh, let me go back to the material. Okay, you should reread 1119 tissues. You should take a look at the handout just so you're prepared for whenever you come across any of those things in your reading that you're ready to go. And this is today. Okay, do you want a break?
I'm good.
Okay, okay. I'm going to go ahead and do this second one. These two for the 24th, no, I'm going to save those. So what I'm going to do is start you on the tissues, the epithelial tissues, and start with the organs. Now, the organs really are models that we use in the regular laboratory. Okay, and you're not going to have trouble with a lot of them because we've done this stuff. Okay, but this is the way that they set it up, the practicum. Okay, and here's the digestive system. Here's the parotid gland. The parotid gland is made of glandular epithelium. Pardon me for saying it again, because I know I have. It secretes salivary amylase for digestion of starch, and it lubricates chewed food. It's an exocrine gland, meaning that it puts exudate or expels a fluid through a tubule. Here's the tubule. It's called the parotid duct. It's also known as Stenson's duct. Here's the submandibular or submaxillary gland with the same function. The sublinguals aren't shown. Please read this material, okay? Because I'll basically be just showing you the slides. Okay, here's the vestibule. The vestibule is the front part of the, in front of the front teeth, but behind the lips. So if you put your toothbrush in there to brush the front, your toothbrush is in the vestibule. Unlabeled is the hard palate and soft palate, but the very end is the uvula, which is useful to making certain speech sounds. I think if we didn't have it, we'd be fine too. The nasal pharynx is above the uvula. Behind the opening of the mouth is the oral pharynx. And then at the opening into the glottis is the laryngopharynx. This is the epiglottis. This is thyroid cartilage. And here's the esophagus. Now, the esophagus is lined by stratified squamous epithelium. The rest of the GI tract is lined by simple columnar epithelium. The esophagus upper portion is lined by stratified squamous. The lower half by simple columnar. All that said here, you've had the word peristalsis from me, so don't. This cat has its trachea showing with the cartilaginous C rings. type of cartilage that those C rings are composed of is called hyaline cartilage. We'll see that on Tuesday. Underneath, so this is pulled up, it looks sort of swirled. That's to get it out from underneath. This is the esophagus. The stomach, we went over this quite recently, but just review the cardiac sphincter here, pyloric sphincter here, fundus, cardia, and pylorus, and rugae in all of the compartments. The duodenum is here. The stomach's not shown. So after the pyloric sphincter, here's the duodenum. These circular folds are the plicae circularis. They're also called circular folds. Here's the pancreas. For reference, here's the spleen, which is composed of reticular tissue. When we talk about the lymphatic system, it's all reticular tissue. The lymph nodes, for instance, reticular tissue. Hard to see unless you've got a nice screen, but here's the gallbladder right here. Back behind it, I think it's in brown, is the liver. This is the gallbladder, emptied by the cystic duct. I can barely see it. I can see it. It's clear enough. But yeah, I don't know if you'll be able to. Hang in there. Okay. Real short cystic duct here. And then here's the common bile duct down here. Up here is the hepatic duct, and it didn't get labeled. The hepatic duct and cystic duct form the common bile duct.
Is the gallbladder usually that vertical?
You've got to take some of these models with a grain of salt. Okay, I think this part of the model is pretty damn good. I think this part of it is diagrammatic. So no, it is not.
Okay.
Okay, now this one would take a while to figure out, maybe. This is the stomach. Well, look around. Oh, that's the small intestine. And that must be then the inferior part of the organism. Here's the thoracic cavity up here. Okay, so here's the stomach. The food's going this way, and it goes out here. Okay, I don't know why this is discolored, or if it is discolored. In fact, this is the duodenum. In the small intestine, in the wall, we have villi. Villi only occur in the small intestine, and they're like projections inward that greatly increase the surface area. The epithelium is simple columnar. This model shows it pretty well as simple columnar. Some of them don't for some reason, but that's the way it is. This simple columnar in the villi is adapted for absorption into the lamina propria. This is the villus. And here's the lacteal. The lacteal absorbs lipids, emulsified lipids that have made it into the small intestine. The contents are called milk. We mentioned that the other day. That's because that's what they look like. Okay. Now I think I'm spouting off most of the reading up here, or I'm confident that you've had it enough times. But if there's anything that you find that you're not sure of when you go through it, just make a note. You can ask me. Okay. Now here, from the small intestine into the large intestine, is the ileocecal valve. And the food first enters really the cecum. We say that, but that we'd have to be standing up. If we're lying down or on our side, it could go in a different direction, of course. But in anatomical position, it would go to the cecum. Here is the appendix. The ascending colon, and you'll notice the haustrum. The plural is haustra. The compartmentalization that helps to move the contents forward. Not labeled. This is the hepatic flexure, transverse colon, splenic flexure, descending colon, sigmoid colon. This white line or gray line and this one are the tineae coli, the three bands of longitudinal smooth muscle that, since they don't contract at the same time, but differently, cause the puckering of the haustra. It may look like there's just one ribbon running through here, but there's not. There are three. At roughly 12 o'clock, 4 o'clock, and 8 o'clock. Okay, another one where you got to look for just a second. Well, you know these are small intestines. Okay. That's either going to be stomach or large intestine. It's large intestine. Here's the end of the small intestine. Here's the ileum. The ileocecal valve is right here. The cecum is over here. And the large intestine is here. It's really the ascending colon. The diaphragm here, the upper body over here, and the lower body, here's the liver, and here's the small little gallbladder. Did I go over this one with you? Does anybody remember me going over this one?
I don't recognize
No.
it, but I also was taking the test.
Okay.
No,
I
you
didn't hear
didn't.
any yeses anyway. So
don't think you
I
did.
don't think huh? I don't think I went over it at all. Okay, I just wanted to be sure I don't go over this one twice. It looks horrible. I mean, it's a nice picture. It just looks like, my God, I'm supposed to know that? Okay, well, this is the liver. This is the liver. And this O is in green. This is the gallbladder. The cystic duct isn't labeled. The hepatic duct isn't labeled. Okay, and this would be the start where my arrow is right now of the common bile duct. We best start right in the middle, right here. That's the incoming hepatic portal vein. It's coming from the small intestine. Small intestine has absorbed material that could be suspicious, and the liver wants to act upon it first. So all the material, all the blood from the small intestine, after it's had a chance to absorb material, comes back through the hepatic portal vein and spreads out throughout the liver. And then it goes into what are called lobules. Lobules. I have a picture of that coming up. It is filtered and then returned to veins. The veins are here. And those veins make their way to this large vessel. This is the inferior vena cava, and it will take the processed blood now to the right atrium. Okay. Over here is the hepatic artery. This was the hepatic duct over here. This is the hepatic artery. That blood coming from the hepatic portal vein is not to nourish the liver. The blood to nourish the liver comes from the hepatic artery. And the blood comes in and spreads out along the red vessels, nourishes the liver at its capillaries, and then comes back by way of veins, the same veins used by the hepatic portal system, and they return to the inferior vena cava. So there's two systems going on in here. One, just regular systemic circulation. That was the last one. And the first one is the hepatic portal circulation. This strip right here is to represent the falciform ligament. The toughest thing about this picture is what's the orientation. I haven't really figured it out myself. This should be on the right side, and the biggest portion here should be in the front. So if I were going to do this and tell you the orientation, I would take this side here and flip it over to here. Okay, and turn it. Don't worry, you're not going to get asked that. It just, here's a lobule. The lobule is the functional unit of the kidney. What type of epithelium is the kidney made of? Glandular, or not, sorry, kidney, liver. The liver is made of glandular epithelium, the whole thing. Okay. When I don't get an opportunity here. This is a lobule. It looks like a little package of brown swagger. You ever seen them at the store? Here's a lobule, half a lobule here, a full lobule here. Well, the lobules, which are roughly hexagonal, six-sided, come together. They have at each of their 6 points a smaller vessel right here, a smaller area. It's called a triad. Here's the triad over here. Oh, that's much better. This is the triad. And here's the hepatic artery. That is bringing blood to the liver. Here's the portal vein. It's bringing blood from the small intestine to be processed, not to nourish the liver. So the blood comes here, bubbles up, and now starts going down these channels. And the liver cells, the glandular epithelium, detoxify it as best that they can. It's not perfect. And the blood exits by the central vein. We have three things, that's why we call it a triad. A portal vein, hepatic artery, and a bile duct. We know the liver produces bile. This is going to the gallbladder. Liver, largest gland of the body. The only gland that is not composed of glandular epithelium, said it the other day, it's nervous tissue in the neurohypophysis, okay, the anterior pituitary. I'm sorry, posterior pituitary. Okay, here's a sort of a summary of gland types of the digestive system only. The parotid glands are glandular epithelium. Upper esophagus, stratified squamous epithelium. Lower esophagus, simple columnar. Simple columnar for the stomach. Simple columnar for the small intestine. Simple columnar for the large intestine. And finally, the liver and pancreas as an accessory organ. These are both glandular epithelium. Okay. If you're looking for what to read, I would reread the two lectures I just had, short as they were. The one on tissues, the first one on tissues for today's date, and then the one that I just did for the organs to get started on. There is a lot of material in this section. If you want to get ahead, you can go to my website again. Oh, I put it away and then I hit it. Here it is. And you can go down to Unit 4 and begin looking through the material there. That's just to get ahead. That's not necessary. Keeping up is most important. Okay, anybody with the last question? Because I'm going to end it short today.
I think I'm all good. Thank you, Charlie.
Okay,
Thank you.
Thank you. Bye.
okay.
Have a
I'll
good
wait
day,
around for
Charlie.
any questions if you want to do anything in private.
Thank you, Charlie.
Bye-bye, Jaden.
Bye, Charlie.
Bye-bye.
All right. Have a good night.
Okay.
Charlie, I have a quick question. Are you able to check my grades, like, to see if I'm missing anything? I just want to like make sure I'm on top of.
Yeah, yeah. Just hang in there for a moment, okay? I'll get
Alright.
right back to you.
Yeah.
Hey, Charlie, it looks like the class just finished, unfortunately. I was having internet issues at home and I just got on and
And this is the day we finished early.
oh man, I was thinking I might be able to make it, but this is just
It's recorded.
okay. I'll definitely be checking that out.
That's won't be posted until tonight. Takes them a few hours to process the recording, but
Okay.
it'll be up. You can see it.
Thanks, Charlie. I appreciate it. Glad I at least caught you.
Okay.
Have a good rest of your evening.
Bye, Tyler. Emilio,
Yep.
you're fine.
All right, perfect. That's all I just wanted to know. Thank you very much.
Okay.
Have a good night, or have a good day, Charlie.
You too.